Okay, this is an anaerobic growth box. There's a gas pack in there that removes oxygen, and the growth medium is Brewer's Agar, and the oxygen indicator is Resazurin. So if we pulled it out of the box, it would turn bluish. This is a confluent lawn that was exposed to 3 minutes of UV light with a card over it. So here's the live part and here's the dead part, and it died because of the UV radiation. This is MS on agar, Micrococcus magmatis, and the top of it is dry and crusty like dead mummy skin. Here is E. coli, and it is growing in punctiliform tiny polka dots, and then there's that nice big globular growth there. And then this is Micrococcus luteus for contrast, which might look similar to E. coli, but ML, the giveaway is that it is canary yellow. Okay, here is nutrient gel, so we can see if it's digesting the gel. I'm jiggling it. MS and E. coli do not digest the gel. Pseudomonas and Bacillus cereus both do. You can tell the difference between Pseudomonas and Bacillus cereus because the Pseudomonas is greeny purpley. And you really can't tell the difference. Oh, on the MS there's growth on the top, and on the EC there's no growth on the top. So that is the difference. Here is Hectoin enteric. The olive blackness is Salmonella. If it were peachy, fleshy, orangey, it would be either Enterobacter or E. coli. Okay, this is rose agar, which determines hemolysis. On the left side is alpha hemolysis, which is partial hemolysis. You can tell because it's kind of greeny. And then on the right-hand side, this is supposed to be beta hemolysis, and that will have a clear halo around the microbe because it will have eaten the blood in the agar. Okay, here is confluent lawn, and there's a zone of inhibition. You will note that the ammonia has a bigger zone of inhibition than the alcohol because it's a more effective killer. And then here's some confluent lawns with zones of inhibition from the antibiotic things. This plate is green. This is Hectoin enteric. This plate is very rosy red. This is rose, and it's labeled, so that one's a gimme. This is Brewer's Agar, which is the oxygen sensing one. This was Eocene methylene blue because it's red, but when you hold it sideways, it's got a bluey line. This one is mannitol salt because it's kind of a peachier red. And this one is McConkey, because when you hold it sideways, it's a bluer red, and when you put it like that, it's a pinker red. Here is Brewer's Agar. In aerobic conditions, 3 grew Micrococcus luteus, E. coli, and Bacillus cereus. And then in anaerobic conditions, only the Clostridium sporogenus grew. And the Brewer's Agar changes color if it is exposed to oxygen. Here is Pseudomonas growing on TSA agar. You'll notice that the Pseudomonas turned it greeny purpley. Okay, on the left is Micrococcus luteus because it's nice and yellow, and on the right is Serratia marcescens, grown at room temperature because it's nice and red. So on the left is E. coli on McConkey agar, and on the right is Salmonella. This one is producing alkali, and this one is producing acid. Damn it.
Opposite.
This one is producing acid because it's pink, and this one is producing alkali because it's yellow.
That's right. What's the purple? Which purple one?
Bloop.
This one.
This is nutrient gel with Pseudomonas, which has dissolved the nutrient gel on the left, and this is nutrient gel with E. coli on the right. It did not dissolve the nutrient gel.
Yeah, that
So
one
I'm
is
wiggling it, you
actually
can see that the
a
Pseudomonas
mat,
side is
and it's,
dissolved.
Tuesday, so
This is
I wouldn't
Clostridium
expect it to
sporogenus,
show
which has
its
produced some
behavior
gas
yet.
at the bottom.
So that's what these are. These will be hemolysis. I'm hoping
This is Staphylococcus aureus in the motility medium with TTC. It has not moved.
What's the difference between these?
And then it has that color indicator.
This is Pseudomonas, which is a moving thing in motility medium with TTC. This is not Serratia.
That's right. What causes the green pigmentation.
Pyocyanin pigmentation causes the greenish, purplinish of Pseudomonas. This is Pseudomonas because it's greeny purpley.
Yeah, but what,
On the left is
It produced a gas and what it was.
Micrococcus
to
luteus
say what-
because it's yellow, and on the
you wouldn't
right
have to
is
say what organism it is
Mycobacterium.
right now.
because it's
we haven't really
crusty
done a
dead
lot of
skin.
testing. Those
Is
are the motility
Micrococcus luteus, which is dry and flaky like dead skin.
Take your notes. What's this one? What about this one? What's happening here?
Okay.
That one, of course, is Serratia marcescens.